All right, so the first part of today is kind of a review of what we were doing yesterday. And so what I want to start with is I want to go back through this idea of translation using this animation simulation. And then I'm going to give you some time to work together and try to kind of stumble your way through, you know, kind of the whole process, transcription, translation. And so I'll give you a website to go to and some time to kind of click through it, work out some of the kinks and just try your best to do what you can with it. So we're starting with translation here, and we've already seen when we get to the translation process, this transcription process that is on the screen, that's already taken place. That was what we spent the bulk of time talking about yesterday, where we're making a replica copy of the DNA using messenger RNA. And that's the transcription process. In the transcription process, that messenger RNA travels from the nucleus to the cytoplasm, that is the area outside of the nucleus, the wet aqueous part. And here's where the protein building is going to actually take place. In this translation step, we're going to read that messenger RNA strand, and we're going to link together amino acids to create this protein. Now, the majority of this process of translation takes place on these. These are called ribosomes. Ribosomes are one of the organelles inside of every cell, and its primary job is building and creating proteins, pulling together amino acids to translate these RNA sequences into viable proteins. Now, in order to do that, you have to have available amino acids. And those amino acids are kind of just floating around in that cytoplasm. So looking again, here is our messenger RNA strand. We can see the individual nucleotides that make up that strand. And what happens is that the ribosome is going to interpret these messenger RNA sequences 3 nucleotides at a time. This is what we have been calling a codon. With the codon, the ribosome can read a particular sequence. And again, going back to our codon key, we can pull this up and say, okay, AUG, that is the start codon. So this is the part of the sequence that the ribosome would be looking for and saying, okay, start translating from here. So AUG was the first part. That's the start codon. Tells us where to start building the chain. As it's starting to build the chain, now we start to see these anticodons come in. And these anticodons are going to attach to the messenger RNA and help link together the amino acids, because they're all going to sit on that transfer RNA. So the transfer RNA has an anticodon, so it can attach to the messenger RNA. And on the opposite side, it's going to have an amino acid that it can use to build the protein. Now, we talked about this on yesterday. Transfer RNA is basically a RNA strand that has been coiled up on itself to such a degree that we actually get hydrogen bonding to occur between the strands to kind of create this weird coiled up shape. but here it is, it attaches. The anticodon attaches to the codon, and we've got the amino acid here ready to go. The ribosome moves to the next codon, attaches the next anticodon to the sequence. And what we see here, we've got another amino acid. This time we've got CGU. That's going to give us arginine. What happens in this process, those two amino acids are now close enough together, they can form a peptide bond. The peptide bond forms between the amino acids. And now, because the amino acid chain has started to being built, this transfer RNA is going to detach itself from the messenger RNA and leave behind the amino acid, just like that. So now the ribosome moves on to the next codon. Next codon comes in. We make another peptide bond. Peptide bond forms. Transfer RNA drops off. And again, until we hit one of those stop codons. When we hit a stop codon, that is the chemical marker to tell the ribosome that it can detach. When it detaches, this last transfer RNA detaches from the amino acid chain as well. And that amino acid chain then can go through the processes that we talked about last week as far as proteins go. That is the primary structure of the protein. It can fold up on itself, it can twist up on itself, create a secondary structure, a tertiary structure. It can do whatever is necessary to become the functional protein it's intended to be. And so here we have an example of what that 3D structure might look like. Now, one of the cool things about protein formation is that there are multiple ribosomes inside of your cytoplasm, and they can code the messenger RNA multiple times simultaneously. So if your body needs to go through the process of creating it functionally a large quantity of a given protein, that same messenger RNA can be used to create multiple strands of the same amino acid chain at the same time. So in this particular example, you've got 4 ribosomes that are coding and translating that messenger RNA at the same time. As they reach the stop codon, they're each dropping off their amino acid chain. Those amino acid chains can then fold up on top of themselves, create a tertiary structure, or combine with each other to make a quaternary structure, depending upon what the protein needs to do to become functional. All right, so what I want you to work on right now, you can get together with a couple of people if you're most comfortable doing that, or you can access this directly. So where I want you to go is this University of Utah website. It's a very simple website to access. If you have a computer on hand or a tablet, you can access it directly that way. If you are just holding a phone right now, QR code will take you to that same website as well. But what you want to do, go ahead and go there. Let me load it up here as well. And I'll put that up. So one way or the other, go ahead and get to that website. Now, I like this particular interactive a lot.
Transcription and translation are the process.
It's an integrated mix of videos and activities. Now, if you don't want to go through the videos themselves, because we've already kind of talked about them here, that's completely fine. You can fast forward to the very end. And then what it'll lead you into is you've got the ability to choose a gene, so part of the genetic code. And it's going to walk through the processes of transcription. And so here we have a bit of genetic code that we are going to transcribe. So we need to put in the correct amino acids to do so, or excuse me, the correct nucleotide groups to do so. So you'll click your way through those. And then it'll lead you into a translation exercise as well. So if you want to skip through the videos and just not do those, just get into the interactives, that's perfectly fine. But I'm going to give you some time to kind of play around with this and just kind of work on kind of those skills of recognizing the pairs and picking the correct kinds of combinations to do the transcription and translation. So give you about, let's say 20 minutes. If we're done sooner, we'll go that way. But if not, we'll cap things up at the 20-minute mark here. All right. So how was that activity? Help a little bit at least? I liked it a lot. I originally had a much more complicated simulator, and it was like biochem kind of talk, and it didn't work as well. So I'm glad I ran into that. That helped a lot for just kind of, you know, giving me a better picture than what I could even give you out of the textbook. So you know, those are there. They're always there. Feel free to come back to those at any time, especially as if you get stuck in your studying. We've got one more thing to do, and that's talk about, well, what happens if the DNA or the RNA sequence gets modified somehow. This is the topic of what are called mutations. So genetic mutations take place when the DNA or the RNA gets corrupted at some level. And it can have some really interesting kinds of effects. So most of us are familiar with the albinism, the creation of albino species or albino traits. So here you have an albino peacock. Yes, Sean. I have a question. Okay, so is that piebald? Is that I'm sorry? Piebald. I'm not familiar. Okay. I would assume so. Albinism is something that is related to genetic mutations. And in particular, it's related to the production of melanin, which is what causes pigmentation. So in humans, we usually think of melanin as being the thing that pigments our skin, pigments our hair. But obviously that can extend to other areas other than just shades of brown and red, which in the case of this peacock would mean that its ability to create the blue and green and yellow feathers completely goes away as well. So mutations are changes in the sequence of DNA. And when we see these changes in the DNA, we know that that is going to affect, obviously, if the DNA is replicated, we're going to get more of that genetic mutation. Or when it's transcribed into messenger RNA, it's going to just transcribe into a different set of codes. And as a result of those different codes, thinking about things from this transcription and translation kind of idea, we know that if we change the code, it could very well change the amino acid that gets put onto the chain. And if that is the case, that's obviously going to affect the structure of the protein that forms and its ability to function properly. Now, we can get these mutations in a lot of different ways. X-rays have been known to cause mutations. Overexposure to sunlight. Since sunlight is, at least the most dangerous part is the ultraviolet rays. Ultraviolet light, higher energy light, is able to disrupt the sequence of our DNA as well. Exposure to certain types of chemicals. These are chemicals known as mutagens. They can have a particular impact on your DNA. And even certain kinds of viruses can have an impact and change your genetic structure. Depending upon where these mutations occur, it can lead to some different kinds of things. So if the mutation occurs in a somatic cell, that is one of your body cells, not one of your reproductive cells, then the altered DNA really only impacts that individual strand and any cells that might be created off of that strand. So as that DNA replicates and makes new cells, those are the only ones that are impacted directly. Now, the issue there is that if those cells happen to reproduce and divide at a rapid rate, this can become the basis for cancer. A rapid growth in cell replication as a result of a mutation that can lead to a mass of cells, and we're talking about potential tumors, both the malignant kind and the benign kind, so the bad kind and the good, or at least the not harmful. If it occurs in a germ cell, so that is, if you're a woman, your egg cells, if you're a man, your sperm cells, then that mutation is going to happen in anything that egg or sperm kind can interact with. So if it is a function of your reproductive organs having issues, especially for men, since men generate many sperm a day, if a mutation happens in the genesis area zone, all of the sperm could end up with that mutation. If it's just an individual sperm, wouldn't really make a difference unless that sperm happened to fertilize an egg and conception take place. And then the new individual would carry that mutation. And it would be in all of their cells because it's starting with a mutated cell. Now, there are different types of mutations that are possible. Probably the most common one is something called a point mutation or a substitution, where in the process of being exposed to whatever it is that we're exposed to, or whatever happens to cause the mutation, one of the base groups gets exchanged for another. We take it out an A and we put in a C. Or any combination thereof. Well, when that happens, one of two things can happen. If the resulting translation results in a new amino acid being put on, then what we have is a missense mutation. If we happen to get a mutation that actually doesn't yield anything different, then it's called a silent mutation. So what we're talking about in those silent mutations is we know that certain amino acids might have 3 or 4 different codon codes that will write for it. If we go from one of them to another one that codes for the exact same amino acid, even though there was a mutation inside of the DNA, we wouldn't actually see it. That's why it's called silent. If the mutation changes a stop codon into an active codon, this is called, let me rephrase. If it changes our sequence from an active codon to a stop codon, then it's called a nonsense mutation. Basically, it cuts it too early. So maybe the amino acid sequence is supposed to be 100 long, but the stop codon happens at 50 instead of 100. The protein is now incomplete and probably useless. That's why it's called a nonsense mutation. The protein that is generated is nonsensical, useless, can't be done with. The last type of mutation here is a frame shift mutation. And this is where we see either an insertion or a deletion from the code. So an extra nucleotide gets added, or one gets taken away. And as a result, the coding gets shifted. Because now you have an extra, and so we end up coding in the wrong direction. We end up coding different amino acids than we're supposed to because of that extra piece in there. And we get to the end of the sequence, and there aren't enough codon or not enough amino acids to do an additional codon. It kind of just stops in place there. These are called frame shift because they think about Scantron. How many of you have ever done this? You got 1 through 20 done. You go to do number 21, but you put it on number 22. And every single answer from 22 down to 57 is wrong because you skipped one on the way. That's a frame shift mutation. You inserted an extra line, and it made everything else wrong afterwards. It would be just like you skip a problem, and instead of putting in the bubble for number 21, you put in the number 20. Everything would be wrong afterwards for the exact same reason. Go ahead.
So, this, but would each of those mutations cause something, or could they all, like, could all of those cause cancer? Or would it just be like a frame shift mutation that would be more likely to?
It would depend. So, obviously, a silent mutation isn't going to do anything. Depending upon what happens, so like a nonsense mutation generally is not going to lead to cancer necessarily, because what's going to happen is you're going to end up making an incomplete protein. So it is most of the time it's non-functional. Now that could have other kinds of impacts on your health, depending upon what that protein's job was, but it wouldn't necessarily lead to cancer or something along those lines. Usually and we'll talk about some more specific kinds of cases. But yes, usually if we're seeing some kind of genetic modification, doesn't always lead to cancer. There are other kinds of health conditions, genetic conditions that could result as well. But yes, assuming that whatever it is functional, then it would have cancer potential. So here's a picture, or at least an illustration, of what normal transcription and protein synthesis would look like. So we've gone through this process already. You got the DNA. The DNA gets transcribed into messenger RNA. Messenger RNA gets translated into the amino acids that make up a protein. And there's usually a very defined order that goes on here. But when we have mutations take place, here's an example of a substitution. So let's say that just this one nucleotide was changed. So this was a C, now it's a T. How does that change things? Well, the stop codon didn't get changed. We're going to have the same 4 amino acid sequence. But that change there is going to move us from glycine to serine. And if you go to glycine is a nonpolar amino acid. Serine is a polar amino acid. That change in the sequence is going to have some pretty interesting and negative impacts in terms of how that protein is able to fold on top of itself to make that tertiary structure. Because there's a big difference between polar and non-polar in terms of how they're going to be able to interact. So that's just a single mutation. This is a point mutation. Substitution. Maybe only changes one of the amino acids, but maybe that's enough to do something noticeable. In a frame shift mutation, again, we're looking at the same sequence. So the sequence here gave us 4 amino acids and the stop codon. So stop codon said, okay, stop the synthesis right here, we're done. This frame shift mutation where we lost a base along the way. Now our messenger RNA is short. There should be 15 base groups here. But now because of that loss, there are only 14. I don't have a stop codon here, but I also don't have enough base pairs to know what to do next. And so we have two consequences. First of all, our amino acid chain changes because now instead of phenylalanine and serine like it was here, or phenylalanine and glycine like it was here, now we've got leucine and alanine, two completely different base groups. And we don't have a stop codon, which means depending upon what is available, this may not stop coding. Or it might just end up sitting here kind of waiting for someone to tell it what to do. So here's kind of the flow chart for what happens when these mutations take place. When DNA gets altered, the net result of that alteration is the creation of a defective protein. And that defective protein can have two consequences. I guess three. One is that it does nothing. But the other two are considerably more impactful. One is genetic disease. So again, that happens in the germ cells. So, talking about eggs or sperm. Then we're talking about genetic diseases because, again, if that germ cell happens to have a genetic mutation in it, and it is able to pair up with its corresponding germ cell, male or female, the net result is going to be a fertilized egg with a complete genetic picture that is altered. That's going to alter every single other cell that comes from that fertilized egg. Now, in somatic cells, again, not inside of your reproductive system, but anywhere else, that has the potential for cancer. So genetic diseases come as a result of defective enzyme production. And that production of those defective enzymes come from mutations in genetic code. So just a couple of examples. Phenylketonuria, PKU. This happens when DNA cannot direct the synthesis of phenylalanine hydroxylase. So the enzyme that converts phenylalanine to tyrosine. And albinism takes place when the enzyme that converts tyrosine into melanin becomes defective. Now, your book has a list on table 17.9 of a bunch of these different kinds of genetic diseases. So in addition to albinism or PKU, we can talk about things like cystic fibrosis, Down syndrome, muscular dystrophy, sickle cell anemia. These are all genetic diseases that are usually inherited. I won't say usually, they're inherited from your parents or from other members of your family tree. Not all of these are things that are in the camp of, if your parents have this, you will get this. And some of them, like Down syndrome. Down syndrome is something that is created by a trisomy. So trisomy, the creation of 3 chromosomes, 21 chromosomes. That is something that is the point mutation. It is a random event. It is not necessarily a factor of, okay, well, the mother has this, so the baby is going to have it as well. So it could have happened on either side. But it would have been a random event. So, you know, you get exposed to X-rays and it affects one of your eggs, and that egg gets fertilized and your baby's born with trisomy 21. Or man gets exposed to a mutagen. It creates a chromosomal duplicity at chromosome 21 for a batch of his sperm, and those sperm end up fertilizing an egg and creating a child, and then the child has Down syndrome as a result. It's not everything or nothing kind of thing. These can be random kinds of things, and at some level, it almost becomes a matter of kind of just dumb luck. And that's why the statistics are there. This happens about 1 out of every 800 live births. Now, other types of genetic abnormalities like this do exist. So a trisomy is not exclusive to chromosome 21. It just happens to be that it's a trisomy that does not result in fatality. So in other chromosomal areas, a trisomy like that might just lead to a pregnancy that's not viable. The mother miscarries early in the pregnancy because genetically the child couldn't survive. And the only way to really know that would be to do genetic testing. And even at that, genetic testing in that first trimester is very, very fuzzy. It's not super accurate. No. I mean, this gets into another kind of moral debate, because a lot of doctors' offices now will encourage genetic testing in the first trimester to try to pinpoint potential issues. The question ultimately kind of falls into, do you want to know that kind of information? And if you did, would you do something about it if you knew? And I'm not going to twist us into that debate. That's a very different kind of discussion than the one we're having right now. But the thing to know about that is the genetic testing in that part of the pregnancies, it's not super accurate. And so you would always run the risk of, if they identified a genetic abnormality and it wasn't actually there, and you decided to end the pregnancy as a result of that information, that's something that, you know, personally you have to keep on your conscience, and you just want to know. Sean?
I forget
No, I didn't, but.
No, forget what it is. It's some kind of like. No, I don't.
All right, let's let's go ahead and wrap this here while Sean looks that up. Okay, so 3 different types of mutations. Well, I guess we'll break what? Bloom? Okay. Okay. What type of mutation is this? If cytosine just enters the sequence. This is a frame shift, and specifically it is an insertion. All right, what about letter B? Adenosine is taken out of the sequence. Yeah, this is also a frame shift. And this time it is a deletion. And if the base sequence in DNA goes from TGA to TAA, this is a substitution. And so another name for substitution is point, a point mutation. So the question is, would it actually make a difference? Well, that's where we're gonna have to go to our key here. We would be looking at in the RNA, ACU. See, ACU correlates to threonine. AUU is isoleucine. So this would be, yes, this would be a missense mutation because we are going to get a different amino acid as a result. All right, any questions before we put this up? All right, so that is the end of chapter 17. And so